Konnichiwa guys, welcome to our channel Loops of Biology. I am Yogiraj Jakkal taking you all to the next part of Brainstorm. As we have completed and wrap up other part of protein, we will be moving with a brainstorm questions alongside the concepts of carbohydrate, lipids and proteins together. So whenever I am considering my first question, it will be if a Ramesh is provided with a sample A and B and wants to find the presence of sugar in both, alright, the Ramesh has to find the presence of sugar in both the sample, whether the sugar is present in sample A or B or it is present in both. What can be the possible way? Set the experiment using the respective apparatus, that is a Petri plate, bacterial colonies and incubator. That is something interesting. We have learned about the reducing sugar and non-reducing sugar. If we want to suppose decide non-reducing sugar to differentiate from reducing sugar, so non-reducing and reducing sugar can be differentiated. Reducing sugars can be differentiated using Benedict solution. Right? There was a Benedict solution containing copper ions. So that was helpful for deciding or differentiating your non-reducing and reducing sugar. But what if both the sample contain non-reducing or both the sample contains reducing sugar? That will be a problematic case. So if suppose sample A, we don't know whether the sample A contains sugar or not, and there is a sample B. So I basically taken a Petri plate, and differentiated this with two different colonies of bacteria. So suppose I am taking one bacterial colony for sample A and another bacterial colony for sample B. Easy. So after this, I am adding sample A and sample B on this particular bacterial colony and keeping this colony inside incubation. So I incubated this particular plants. Now, after incubation and providing them with the optimum condition, this bacterial colony will start growing and utilizing the sugar present inside them. If there is a presence of sugar, it will start showing a process of fermentation. And this fermentation will give you an acidic smell. There will be an acidic smell and effervescence. So what do you mean by effervescence? That is nothing but a bubbly structure over the surface of this bacterial colonies. So what are the medium you are going to take? That is nothing but either sample A or B. Based on the presence of sugar, there will be an acidic smell and effervescence coming out from that particular sample. And that will indicate the presence of sugar present inside a sample. So suppose if sample A is providing effervescence, then sample A must be having sugar, and sample B, if not showing effervescence, suppose sample B is not undergoing fermentation, then it does not contain a presence of sugar. I hope you cleared this concept and understood the logic behind it. Whenever I am considering any living organism to differentiate between the presence of sugar or absence of sugar, then I have to add that particular sample on that bacterial colonies. Then I have to incubate them for optimum condition, and after the growth, If they utilize the particular sugar inside them, then that sugar will undergo fermentation, and finally the acidic smell and effervescence will give you the positive result for the presence of sugar. So next question, a ram has given a glucose after a long run while Sita was provided with fructose and Varun was provided with milk. Then who will feel more energetic first and why? Okay, so let's consider Ram, who was being provided with glucose. There was Sita, who was being provided with fructose, and there is Varun, who has been provided by milk. So you can eliminate one option from this. How? If I'm considering glucose and fructose, both belongs to your monosaccharide group. Yeah, I know the fructose belongs to ketosis while your glucose belongs to aldoses. That doesn't matter over here. They both are monosaccharide. So option should be Ram or Sita. Alright. Why not milk? The milk contains lactose. And this lactose is basically a disaccharide. Whenever a disaccharide is provided to any individual after a long run, then that particular disaccharide has to be broken down into monosaccharide for easy absorption of molecule inside the blood. For absorption in blood, and finally obtaining energy by transferring that particular molecule inside a cell, that is called as assimilation. So there is for absorption in blood to assimilate. Now which disaccharide? Alright, the lactose is made up of what components? So the lactose was made up of glucose plus galactose. So we will get two monosaccharide, but still we have to break down first that particular lactose as disaccharide into a monosaccharide and then obtain the complete energy. While if we are considering Ram and Sita, they are provided with direct monosaccharide. So this is basically easily absorbed in the blood. Now, if both are getting easily absorbed, then which one should be preferred and why? If I am considering glucose, suppose there is a glucose which is transported inside a cell. Alright, and there is going to be a fructose which is transported inside a cell. Now, both are going to give me the energy, and through which pathway? There is a universal pathway called glycolysis. Alright, so they have to break down glucose through glycolysis. Whether it can be glucose or fructose, both has to enter inside the glycolysis itself. But whenever the glycolysis starts, it starts from glucose. And we can skip 2 steps to enter directly inside a fructose if we are providing fructose inside a cell. So whenever I am providing fructose directly inside a cell, it is skipping. The fructose skips first 2 steps of glycolysis. And as the steps are going to be skipped, the energy obtaining from fructose will be quicker as compared to the glucose. The glucose starts from initial step. It starts from step 1. Step 1. That's why the fructose is more preferred for faster energy source whenever a particular athlete is running for a long time. So that's why your Sita will feel energetic first because fructose skips some steps of glycolysis. Obviously, if you want to feel energetic by skipping some of the steps or by skipping some of the we can say extra work, some of the extra work, then you will prefer that. So in that particular case, whenever the fructose is provided, it is skipping first two steps of the complete process and then entering inside a process for energy provision. So it is providing energy, but by skipping first two steps. That's why the fructose is more preferable component than that of glucose. Next case, if the number of unsaturated fatty acids are increased in cell membrane, then what will happen to the cell and why? If I am considering unsaturated fatty acids, you must remember whenever I am considering cell membrane with unsaturated with unsaturated fatty acids, then those are basically having presence of double bond, and that will be present between your carbon atoms. So wherever the double bond is present, it is going to form a kink. So this unsaturated fatty acid forms kink in the structure. Now that particular broken leg of fatty acid, now the broken leg of fatty acid is now breaking one more time if the number of unsaturations are increased. So suppose if I am increasing number of unsaturations, then the number of kinks are also increased, and that is because which your cell membrane becomes more fluidic. As the fluidic cell membrane is going to be there, it cannot maintain the shape of cell. As the fluidic membrane is forming, first of all, it cannot maintain the shape of cell. Now what will be the another problem? If I am exerting an extra pressure on this particular fluidic cell, then there will be, if I am considering, or we can consider the Pascal's law of physics. What does Pascal law means? That every fluid is applying pressure inside a cell equally. So fluids apply pressure inside the cell on walls equally. And if the cell membrane is more fluidic, then it can rupture. So the cell membrane can rupture and could not gain its proper shape. So that is one of the main problem that will cause if unsaturated fatty acids are increased inside a cell. Got it, guys? Let's move on to the next question. If Rahul wants to know about structural pattern, okay, so Rahul is decided to know about structural pattern of antibody. Now, antibody are of many types. So, Rahul was provided with IgG antibody with 4 disulfide bond. Let's write down some information. So, there is IgG antibody with 4 disulfide bonds. So if I'm considering 4 disulfide bond, there has to be some chains present inside them. So from this, I can assure that there is going to be presence of different chains. Different chains. And as we have seen that antibody is basically belonging to protein. So chains of protein is going to be present. Then what experiment should he set? Now he has to understand the structural pattern which chains are present. So basically structural pattern is going to decide the molecular weight of antibody plus number of chains, the number of chains and plus types of chains. This is nothing but my structural pattern that Rahul wants to know about. So now he was provided with polyacrylamide gel and beta mercaptoethanol. Whenever I am considering polyacrylamide gel, so this gel is basically forming a pore inside it. So there is based on concentration, the gel forms different diameter hollow pores. Alright. So based on concentration means what? Whenever I'm increasing the concentration, the diameter of that pore is going to be decreased. Whenever I'm increasing the concentration, the diameter of pore is basically decreased. So that means the concentration is inversely proportional to the diameter. Now, what does concentration means? That means number of ions per unit volume. How many number of ions or number of moles are present there per unit volume is nothing but my concentration. So whenever I am increasing the concentration, the diameter of hollow pore is going to be decreased. And through this particular pore, through this hollow pore, the strands of protein moves. The strands of protein moves. All right. Now the next element is given with us, that is beta mercaptoethanol. So that is BME. This BME helps to break disulfide bonds. This is going to help to break disulfide bonds. Whenever I am going to break the disulfide bonds, that is going to separate all the strands of protein present inside it, right? So this breaking helps to separate protein strands of complete structure. Now, as soon as I'm considering IgG antibody, there are 4 disulfide bonds. So if I'm considering a normal structure of antibody, so this looks like something this. So there are 2 disulfide bond here, one over here and one over here. So there is one heavy chain and a light chain present inside the antibody. So whenever I am adding beta mercaptoethanol inside a solution, this disulfide bond is going to get broken down and the light and heavy chains of this antibody will separate out. So based on their molecular weight, we are using polyacrylamide gel inside a method called as native page. Native polyacrylamide gel electrophoresis. So in the presence of native page, we can separate heavy and light chains of IgG based on molecular weight. And depending on the number of bands, if suppose the gel is like this, so there is, if suppose gel is like this, then there is going to be, suppose if this is your one band, this band represents heavy strand, while the band coming at bottom will represent your light strand. So if there are two bands, we can at least interpret that IgG antibody will have one heavy strand present inside it and one light strand. So basically there are two type of chains present inside a complete structure. Now if we have to go for a number, then we can just estimate that particular number based on intensity of band. So molecular weight, we can do that using your page. For type of chain, we can use number of bands, and for the number of chains, we can use intensity of that particular band. So I know this was one of the trickiest question and it was quite tedious to understand. But if you have any doubt with this question, please comment down inside the comment box. So next case will be, how do lipids and proteins interact with each other inside the cell membrane? So if I am considering there are some proteins and they have said inside the membrane, so we can remember this particular part like there was your membrane containing two parts as hydrophilic and hydrophobic part, and there was a protein embedded inside it. So if I want to talk about this particular part which is inside the membrane, the protein will have the elements which are non-polar in nature, That means the protein should have presence of positively charged amino acid, which were arginine, lysine and histidine. So there is going to be either arginine, lysine or histidine present inside that particular part, or the non-polar amino acid can be seen. The non-polar amino acid can be used. So if the protein have to interact with this particular lipids which is present inside the membrane, like this particular part, so they have to show hydrophobic interactions. because even this protein part, so lipids and proteins are designed in such a way that they are basically moving away from water, moving away from water. So if both have the same destination to go, if both are having the same property of hydrophobic, then why don't we go together, right? So in that particular case, whenever the protein and lipids are having hydrophobic nature, they are undergoing hydrophobic interaction through non-polar regions of structure. So wherever the non-polar region is present, that particular hydrophobic interactions are making them stable and settling them within the cell membrane. So in this particular case, how they are interacting through hydrophobic interactions, but that is inside a cell membrane. If we are talking about this upper part will be having negatively charged amino acid, negatively charged amino acid, which can form hydrogen bond, which can form hydrogen bond. So depending on the region of protein, the amino acid are set in such a way that they can be able to interact with the molecule present in the surrounding and remain stable inside the structure they are belonging to. So this was your last question for this brainstorm. I hope you loved this video and you understood all the logic behind the brainstorm questions. Please do not forget to like, share and subscribe to our channel guys, and hope you are loving our video. See you in the next video. Bye bye guys.